Hello, everyone. Thank you for joining us for CNE News' second annual virtual symposium, Advances in Drug Discovery and Development. I'm Stu Borman, Senior Correspondent at CNE News, and I'm delighted to introduce our next speaker, Vic Myer, Chief Technology Officer at Editas Medicine. Vic Myer earned a BS in biochemistry from Cornell University and a PhD in molecular biophysics and biochemistry from Yale University, and then served as a postdoctoral research associate at MIT's Whitehead Institute for Biomedical Research. He held various roles at Corning and was senior scientist for Millennium Pharmaceuticals. He was a founding scientist and group leader at Excelli, a systems biology company focusing on commercialization of a high-throughput cell-based microarray technology. About a decade ago, he joined the Novartis Institutes for Biomedical Research as a research investigator. He later headed Novartis's high-throughput biology and target discovery technologies, and most recently served as executive director and Cambridge site head for the Developmental and Molecular Pathways Department, where he led a team of over 200 scientists. He joined Editas Medicine earlier this year. Vic, please go ahead.
Thank you for the introduction, Stu, and I'd like to thank the organizers for inviting me to present today. It's a real privilege, and I think it's a really interesting platform for sharing information in the scientific community from the comfort of your own office and your own desk. Although I have to say, on a personal note, it's very bizarre to be speaking to my monitor right now. And certainly the folks in the hallway who have been walking by are asking all sorts of questions, so it's a bit odd. I'd say this presentation is a logical follow-on from a presentation that was given by Feng Zhang, who's one of the founders of Editas Medicine. Feng presented, I think, last year in this forum and gave a really nice overview of the CRISPR-Cas9 technology that the company is founded on, and really sort of, I think, highlighted a lot of the biological applications and uses for the technology. And I think, as most of you who've dialed in probably know, CRISPR-Cas9 is really creating a revolution in the world of biology. It's sort of swept through the research and discovery areas like very few technologies have before. And I do honestly think it's one of those sort of once in a lifetime or once every decade sort of biology discoveries. I'm not going to talk much at all about CRISPR-Cas9, the science of the specific endonuclease today. I'm going to talk about what we're doing at Editas, which is working to translate this biology revolution into what we hope is going to be a therapeutic revolution. So the mission of the company is to create a novel class of therapeutics that precisely correct the underlying cause of a broad range of genetic diseases. And I think, as many of you know, the ability to correct genetic mutations has long been a goal of medicine. I think since scientists started cloning genes and describing mutations and really understanding that mutations can cause disease, it's been an aspiration for us to be able to correct that. And of course, many of the drugs that are on the market can correct very few of the root cause mutations of the disease. But I think there's really sort of a unique confluence of events that have come together to allow us to aspire to actually try to reach this goal. And there's three things in particular I wanted to highlight. The first, of course, is the advent of sequencing technologies, the application and understanding that we've achieved in human genetics, and really the collection and understanding of phenotypic and clinical data associated with diverse sets of diseases really sets the stage for us to understand the root cause and the drivers of genetics. The second is that the fields of gene therapy and nucleic acid therapies have really come a long way in terms of being able to deliver very complex payloads and very complex cargoes to a diverse set of areas in the body. And that's a lot of the work that's going to help us leverage our technology to generate therapies. And finally, genome editing is not new. We've been going on for a long time, but I think the advent of the CRISPR-Cas9 system and the discovery of the system has really positioned us to rapidly go after and try to solve some of these fundamental problems. So let me just, in one slide, summarize the technology that we're working with primarily at Editas Medicine, which is a protein RNA complex called Cas9, which is part of the CRISPR system. And at its core, it's a double-stranded endonuclease. Cas9 is. On its own, it has very little to no DNA binding and cleavage activity. That DNA binding activity is provided in trans by an RNA molecule. In the iteration that we're using, the technology, it's a single guide RNA molecule. And that guide RNA molecule binds the protein and imparts upon it the DNA binding specificity so that it can find and target the double-stranded portion of the genome that's complementary to the first 5' end of the guide RNA. So originally Cas9 was discovered. Cas9, the CRISPR system, is part of the bacterial adaptive immune system. It's a system where bacteria have evolved to sort of grab onto invading DNA, either from bacteriophage or plasmids. They then take that DNA, they stuff it into their own genome as sort of a storage mechanism. Then this DNA, upon subsequent infection by an invader, is transcribed. In the case of the type 2 CRISPR systems, the RNA is bound by the protein and directs it to cleave the internal, the invading phage or DNA plasmid. So again, it's a programmable nuclease where the specificity is provided by a guide RNA that's in trans to the protein molecule. I have two diagrams here of the molecule. Shown on the upper left is a beautiful crystallographic image that I grabbed from the Anders 2014 Nature paper. Shown in blue, the blue trace is the protein. Shown in green is the guide RNA, and shown in red is the double-stranded target DNA. And on the right is simply a cartoon schematic of the protein where the guide RNA is shown in the bottom, hybridizing to the double-stranded DNA target shown in the middle of the molecule. Importantly, after the cleavage event happens within the cell, the cell's natural DNA repair machinery takes over to repair that cleavage event. And really, there are grossly speaking, two possibilities in terms of how the cell responds. The first is by a process called non-homologous end joining. And this is a relatively efficient but imprecise mechanism where the cell stitches the double-stranded cut back together. So during this process, the cell will often do this in an error-prone manner, and it will create small insertions and deletions at the site of cleavage. And so this can be an incredibly important and efficient way to knock out genes, because if we target the site of cleavage to an open reading frame, the resulting small insertions and deletions will actually disrupt the coding sequence of that gene, causing a loss of function. The other less predominant pathway is called homology-directed repair. And this is the process by which the cell uses a complementary DNA as a template to incorporate a new, essentially reparative piece of DNA. So it's a way we can correct genes as opposed to disrupt them. And it's an important thing to understand for the audience that there are two different pathways we can go down. One which is imprecise and is generally targeted toward knocking out function, and another which is about repairing function. And ultimately, we believe, and I think others would probably agree, that to fully realize the promise of the technology, we're going to need to be able to harness the homology-directed repair pathway. Okay, so within the company, we've created sort of a dynamic relationship between creating medicines and developing the technology. We're a product company, a medicine company, but we're built on a strong technology platform. So the goals for the medicines group is to provide first-in-class therapeutics and also to create indications that will require advancement and development of the technology. While on the technology side, we aspire to be best-in-class in terms of our platform, and we also aspire to invent and create innovation that will expand indication for our genome editing medicines. And we generally think about three questions as we develop and drive our strategy. The first is, what can we do today? So given the state of the art and where we are, what therapies can we work on, and what can we develop with what we have? The second piece is, which disease indications are springboards for expansion within that therapeutic area? And you can imagine as we develop delivery modalities and we develop the ability to edit, create different kinds of edits in different tissues, we can start with treating one disease, and then we can expand within that tissue type or within that therapeutic area. And finally, we think a lot about improvements in technology space and which improvements will most broadly expand our capabilities. And this comes back to really sort of the push-pull of the creation of new medicines and the development of the technology. We're fundamentally a product company, again, built on a strong technology base. Okay, so what I'm going to do over the next two slides is just talk a little bit about the platform and a little bit about how we think about the therapeutics and how we select what we work on. And then I'll walk through two examples of more advanced programs that we're working on. The first is an ex vivo application where we're editing T cells in collaboration with Juno Therapeutics. And the second is an in vivo application where we're editing within the subretinal space of rods and cones of the eye for a inherited optical vision disorder. So in the platform space, we divide the platform into four key areas, or four key facets, if you will. The first is what we call molecular engineering. So this is all the nuts and bolts of the CRISPR-Cas9 system itself. It's the protein, creation of the protein, purification if it needs to be done, chemistry and biochemistry, engineering and evolution of the protein. And similarly with the guide, it's nucleic acid chemistry, it's guide design, it's all the modifications that one can make to enhance function of the guide RNA. It's also where we do all of our lead finding activities, so finding the best combination of enzyme and guide or guides to solve a given problem. And finally, where we do all of our specificity work. So it's obviously an important part of our technology platform. Once we've sort of defined what the package looks like, we then work to think about delivery. And I'll talk a little bit more in detail about delivery, but what I've essentially diagrammed here is a cartoon of a virus and a cartoon of a lipid nanoparticle. And that's simply to remind me to tell you that we're not a delivery company, but we are working with and licensing in delivery technologies that have been developed over the last 20 years to solve nucleic acid and gene augmentation sort of strategies. And so we're not afraid to develop new delivery technologies, but it's not where we're choosing to invest right now. Beyond delivery is this idea of regulation, and I think this is a really important idea to impress on the audience. In contrast to gene augmentation therapies or nucleic acid therapies where you're looking for prolonged expression or prolonged suppression of your target, we actually want to have very tight control over the activity and the pharmacokinetics of our editing enzyme and our editing package. We want to have the package delivered. We want it to do its job, correct the deficit or knock out the gene of interest in the genome, and then disappear. Right, so this is actually in stark contrast to a lot of the other modalities that I mentioned before, gene augmentation or nucleic acid-based repression. But I think as we talk to people, it's relatively straightforward to take something that already works reasonably well, like some of the existing delivery technologies, and actually turn it back down. And so we have a bunch of programs internally to specifically address this. And finally, the last piece in the platform that we are focused on, and this is, I'd say, a strong commitment for the company, is harnessing the ability for the cell to repair the double-stranded break using homology-directed repair as opposed to non-homologous end joining. And again, I think this is going to ultimately be an important component of creating the most value and being able to treat the most patients with the genome editing technology. So I'll shift gears and talk a bit about how we think about diseases and biology. We think about, as we select what we're going to work on, we think about medical need in these four basic categories. The programs in these four basic categories. There's medical need, there's the biology and clinical regulatory components, there's delivery, and then there's the editing technology itself. So we like to work on things that are not currently being addressed adequately by existing therapeutic modalities, and we want to ultimately be able to generate a differentiated product. So if something is potentially treatable by gene augmentation therapy, for example, it might not be the best indication for us. If it happens to be a very large gene that you would have a hard time delivering using conventional delivery technologies, that might be a good place for us to dig in. On the biology side and the clinical side, we really want to be going after diseases where we understand the natural history of the disease, because that will translate into understanding how much editing we need and where we need to have that editing happen. So for example, recessive disorders where a wild-type allele covering a recessive allele, the individual is normal and is a carrier, where a homozygous recessive is affected with the disease, gives us a tip that 50% rescue of one of the alleles as opposed to both of the alleles should be sufficient to restore gene function. We also, of course, need access to patients in order to carry out clinical trials, and we want to be able to have an appropriate treatment window so that we can see efficacy in the clinic in a time frame where we can understand what's happening with our therapy. This is a new technology and it's a new therapeutic modality, and so I think we're gonna learn a lot in the clinic, and so we need to be able to sort of iterate through that process of creating a drug and testing that drug in the clinic. As I mentioned, in delivery space, we would like to use things that are clinically validated and specific for a target cell and tissue type. And we need to be able to work and understand efficiency and specificity requirements and the sort of amount required for the vector and the dose. And finally, on the editing side, we need to go after things that are within reach today while we set up programs to access technologies or access different repairs in the future. We also, for a given disease, need to understand sort of the human genetics of the situation. Are we looking at a high prevalence of a common mutation, or are there many, many mutations spanning a region that we would need to deal with? Okay, so starting to jump into the data. Again, there are two possibilities. On the delivery side, our strategy is to select the right mode for the problem at hand. So when we think about ex vivo cell-based therapies, of course, there are a lot of different methods by which we can deliver. In vivo, we are a bit more restricted. And then and I'm just showing sort of the different formats here. Once we think about a given format, there's of course different ways that can also be delivered. So for example, in vivo, we can either think about systemic delivery, or we can think about localized delivery, et cetera. So just to start, on the ex vivo side of the programs, I've just diagrammed here 4 different possibilities. We can deliver a plasmid. So the basic idea, I should say here, is we would extract cells from the patient. We would edit those cells, and then we would put them back into the patient. So it's essentially an autologous cell therapy-based approach. We could encode the Cas9 protein in the guide RNAs in a plasmid and introduce that plasmid. We could use an all-RNA approach where we encode the Cas9 on a messenger RNA and couple that with a synthetic guide RNA. We could imagine an RNA protein complex approach, an RNP approach, where we pre-complex the guide RNA with the Cas9 enzyme. Or finally, we could think about a viral vector to use ex vivo to deliver a virus that encodes for both the protein and the guide RNAs. So I'm going to talk about the work that we've done with Juno Therapeutics to date. This is about a 3.5-month-old collaboration, but I hope you'd agree that we've seen some exciting results, and the collaboration is advancing quite quickly. So as probably many of you know, Juno is one of the world leaders in using engineered T cells to treat cancer using both the chimeric antigen receptor technology, the CAR technology, as well as the T cell receptor or TCR technology platform. And essentially there is an autologous cell therapy strategy where they will take cells from the blood of patients. They will select the cell type that's required for the therapy and activate that cell type. They'll introduce a gene that adds a new function into that T cell. So it might activate the T cell when the T cell encounters a specific antigen on a tumor or on a cancer cell. They'll then activate those cells and infuse them back into the patient. And the entire idea of the collaboration for us is to integrate gene editing to enhance the T cell function in the ex vivo portion of the process. So we and Juno believe that there are certain targets that we can go after to either knock them out or change them such that the modification of those targets will positively affect the ability of the T cell therapy to remove the cancer from the patient, to essentially cure the patient. Okay, so we started out this program looking into delivery, and we knew we were going to be ex vivo from the start. We looked at a number of different systems, and essentially we needed to have a scalable delivery system that could integrate into their current workflow. We looked at a couple different things, but ultimately settled on the MaxCyte electroporation system. This is a nice system because we can run it at small scale, and it is scalable up to therapeutic uses. It's in our hands, given high transfection efficiency and good cell viability, and I'll walk you through some of that data moving forward. Just shown on the bottom left-hand side is a FACS plot of GERCAT cells, which are a transformed T cell line, where we introduce the messenger RNA that encodes either Cas9, shown in black, or GFP, shown in red, and you can see we get a really nice shift in fluorescence, and we get very high transduction efficiency in the control experiment. So as we started to look into knocking out therapeutically relevant targets, and I should say up front, I apologize, I am masking the names of the targets because we're not ready to disclose the names of the targets that we're working on together, so you'll see them generically called target or target A or target B. I should say all the targets we're working on can be probed using FACS, and so you'll see FACS data in the next several slides. Okay, so back to looking at delivery. The two approaches that we focused on initially were either an all-RNA approach, where we encoded the Cas9 on a messenger RNA and coupled that with a guide RNA and delivered that to the cells via electroporation, or an RNP, where we pre-complex the RNA with the protein. And the results of this are shown on the left-hand side. So this is just a time course of GERCAT cells that are treated with either the all-RNA formulation in the lighter shade of orange and the RNP formulation in the darker orange, dark red color. And what you can just see is we get pretty good percent editing. I should mention that this assay is a targeted, on-target assay called T71. So this is a mismatch sensitive endonuclease that cleaves DNA when it's got a mismatch. It's a standard editing assay in the field. And we get pretty good editing for both delivery technologies, between 30 and 50% knockout in the target of interest in this case. And if you look at the toxicity on the right, both of them gave reasonable response in toxicity. So we didn't see a large die-off, and the cells seemed to recover. I would say in this one experiment, the way it's plotted, it looks like the mRNA actually had a lower viability, but this did not stand up in repeat experiments, and we now conclude that both messenger RNA and RNP have equally toxic or basically non-toxic effects on the GERCAT cells at least. Okay, so given this data, we decided to focus on RNP, and I think strategically this makes sense because it also gives us more control over the regulation of the molecule, the kinetics of the molecule. It'll be cleared from the T cells more quickly. In theory, it also allows us to chemically modify the protein if we are interested in doing so. And so now we're moving into primary human T cells. So these are CD4 positive T cells that we are editing with a target of interest, and shown on the top is the FACS plot. So top left is the isotype control. The center is a control guide RNA that's targeting a different gene, and on the top right is a guide RNA that is targeting the target of interest. Those two FACS plots on the top right are represented on the bottom left, looking at target expression, and what you can see is a pretty nice shift. Again, we are probing the target of interest here. Pretty nice shift in the control versus the targeting guide RNA. When we go in with that T71 assay that I mentioned before on the bottom right, we see about 50% editing, which if you look at the FACS plot is actually a little bit confusing because it looks like it's much greater than 50%. So we did go ahead and use a sequencing-based approach to confirm the editing events that we saw, and actually it turned out that we were underestimating the amount of editing using the T71 assay, and this is a common challenge in the field in that if you get a lot of editing and those edits are very common in nature, they will appear as unedited duplexes to the T71 assay because, of course, they perfectly hybridize to each other. So we've moved into Sanger-based sequencing and targeted sequencing for all the subsequent analyses because we were already maxing out the capability of the T71 assay to recognize the editing events. So to make sure this wasn't a one-hit wonder, we've gone after two additional targets. Here the FACS plots are in the other orientation. The top left is a control where we've actually knocked out the target in the previous assay, and you can see it has no effect on this target. And then we knock out target A and target B, and we see reduction in fluorescence in the FACS plot for target A when knocking out target A, and target B when knocking out target B. And this data is summarized on the bottom, and what I'm showing is percent viability on the left-hand side, and so you can see an unedited or the control edited cells have a similar profile as the targeted cells, and on the right-hand side you can see the percent knockdown is measured by sequencing. And so here we see a slightly different kinetics, and we're not sure if that's a biology result or if that's actually a delayed kinetics of editing, meaning the edited cells actually outgrow unedited cells. With target A, which is different than the previous target, we are at about 90% edited cells by day 4, and with target B, we're around 50, high 50s, 60%, and so we're still working on making this a more robust result, if you will. So to confirm that this was not restricted to CD4 positive T cells, we've also done this with CD8 positive T cells, and we get very similar results. You'll see these are the same two targets as shown on the previous slide. I should say the same 3 targets shown on the previous slide. And this is the viability and the editing data. So target A is still the one that's easiest to knock out with the guide that we're using, and target B is less so, but we can work in both CD8 positive and CD4 positive T cells. Finally, we need to start understanding donor-to-donor variability, and so this is an experiment where we went after those same 2 targets in 3 different donors. So this data looks just like the previous data. The only exception is that we have error bars on here, and these are error bars that are derived from different donors done on the same day. So these are essentially true biological replicates in that they are entirely different T cells derived, CD4 positive T cells derived from different donors. And this is encouraging to us because this is across a relatively broad age range, and blood types, etc. So we're continuing to dig in on this data. Okay, so that's where we are with the Juno program. We're really able to edit at 90+% efficiency ex vivo with primary human T cells. I'm quite excited about that. We're working with our partners now to assess the biology function of these edited cells to understand if they're doing the biology that we imagine that they are actually doing. So we're working hard with our partners to do that. Okay, so let's shift gears a bit to an in vivo circumstance. So the disease we're working on here is called Leber Congenital Amaurosis type 10, or LCA10. The collection of Leber's diseases is a heterogeneous group of inherited retinal dystrophies, and it's the leading cause of childhood blindness. There are at least 14 different described genes that can have mutations in those genes that cause this disease. So while it's characterized as a single disease, there's clearly different genetic drivers. LCA10 is a homozygous recessive disorder, so the parents are generally normally vision carriers, and they have a Mendelian relationship in terms of the creating offspring that have that carry two defective alleles of the gene. The gene responsible for LCA10 is a gene called CEP290. CEP290 is a protein that's involved in ciliogenesis, and it's important for maintaining ciliary structure and function, and that's an important component of photoreceptor rod and cone function. So shown on the top right is just a retinal cross-section from a normal person on the top, and you can see in purple highlighted the layer of the layer of rods and cones in the retina, and it's with the central fovea highlighted where you get your high central vision acuity from. And what you can see in the bottom is a cross-section from a CEP290 patient. And what you see is peripheral loss of cones, and then that peripheral loss over time in the patient continues to migrate from the outside in until these patients end up being totally blind by the by their mid-20s, at the age of 30. So children are often born with this disorder. They do have a rapid loss of the rods. Then the cones deteriorate in their adolescence and into their early adulthood, and they end up as completely blind. It's important to note that the central photoreceptors are maintained and are functional, so the goal for us is to target these photoreceptors and correct the mutation in CEP290 so that we can hopefully allow these photoreceptors to survive. Okay, so this is a good place for us to start with a new technology and new therapeutic. There are no therapies available. It's a relatively large gene at 7.5 kb, so it's larger than the packaging limit of the adeno-associated virus. There are proximal endpoints we can do within the clinic, so we can measure visual acuity very accurately within the clinic. We can set up very well internally controlled clinical trials where we can treat one eye and not treat the other eye. The eye, of course, is an immune-privileged organ. AAV has been used in gene augmentation therapies in the clinic, and it's been shown to be safe and effective. And we are currently partnered on this program with the lab of Dr. Bud Tucker at the University of Iowa, and the University of Iowa and Bud Tucker are experts in these sorts of therapeutic approaches. I'll talk a bit about the editing that we're doing, but we're also working with Bud on ex vivo models for both efficacy and toxicity. So in terms of the biology, again, it's a large gene, and intron 26 spans the two exons, exon 26 and 27, and of course, in the course of normal people, we get normal transcription and splicing to get the correct CEP290 protein. The mutation of the most common LCA10 causing mutation is actually a single point mutation within the intron. It's highlighted in blue with the the red star over it, and this actually causes a change in the splicing pattern. So you get creation of a novel splice acceptor, activation of a splice donor, and what happens is you get inclusion of a novel exon of 128 base pairs between exon 26 and exon 27, and this creates an in-frame stop codon, which then creates a truncated non-functional protein. Okay. So this is our editing strategy. We're actually using a dual guide editing strategy. So we're going in and with a double-stranded, two double-stranded endonucleases. So it's one Cas9 molecule with two guides. We're directing those Cas9 molecules to cut on either side of the mutation and essentially remove that mutation. So for us, it's a deep intronic mutation. This is not shown to scale, so I should say this is about a 1 kb chunk that we're cutting out, but it's a pretty large intron, so we're nowhere near the exon intron boundaries. The editing itself doesn't need to be precise. We just need to clip out this 1 kb region. So the idea is we'll cut out this region and we'll eliminate the cryptic splicing pattern, and we'll get correct CEP290 protein expression. The product prototype that we're working with is a subretinal injection of a single AAV vector. This is two different guide RNAs directing the enzyme to the two different cut sites driven by the U6 promoter. And we're using the Staph aureus Cas9. And I should mention the Staph aureus Cas9 is a newly described Cas9 that was characterized in Feng Zhang's lab, and we've done additional work that is in review process right now to publish the sort of in-depth characterization of the Staph aureus Cas9. This is an important enzyme for us because it's over a kb smaller than the Pyogenes Cas9, and therefore it gives us more real estate in the adeno-associated viral vectoring system so that we can incorporate more functionality in that AAV vector. So here's some data. So this is measuring the proportion of knockout of that 1 kb fragment, and these are guide RNA pairs shown across the x-axis with the GFP control on the right-hand side. These are patient-derived fibroblasts, so these are actually very difficult to transfect cell, and so we only get about 10 to 20% deletion. We're assaying this using a digital droplet PCR. A standard assaying system won't work in this case because we're deleting a very large fragment, so we have to go to an alternate readout technology. In control cell lines like HEC293s or U2OSs, we do see between 30 and 50% editing, so we think that this is essentially a transfection challenge that we're facing here. But we felt like it was important to do this in patient-derived fibroblasts to understand if we're actually correcting these mutations. This is the effect of that in the same fibroblasts of doing the editing on messenger RNA expression. So shown on the right-hand side right-hand side are the controls. The messenger RNA amount for the mutant message is in green, and for the wild type is in blue. So in the patients, you do have some wild type protein because you get some normal splicing, but the predominance of splicing is at the cryptic splice site. I should say it's our hypothesis, although we haven't formally proven this, that the mutant messenger RNA is actually less stable than the wild type. So this is a misrepresentation of the level of transcription. This is, of course, a steady state snapshot of the levels of messenger RNA. But what you can see when we perform the editing, and again, we're only editing at between the 10 and 20% range, we're getting a good 2 to 3-fold increase of the wild type amount of messenger RNA. As shown in this slide, and here is the protein expression data. So there's a Western blot on the left and a quantitative Western on the right. We're seeing a good 2-ish fold increase in the amount of protein. Wild type protein, I should comment that this antibody actually only recognizes the C-terminus of the protein, so it would not recognize the mutant form of the CEP290. So, I forgot to mention, I did mention initially that this was a protein that's involved in ciliogenesis, and so this is simply a slide demonstrating that we're building a ciliogenesis assay out of these same patient fibroblasts, and shown in it's a high content imaging assay where we're staining with 3 colors, DAPI for nuclei, acetylated tubulin, and gamma tubulin for visualizing the cilia. You can see in the top left blow up a nice ciliar formation with the red acetylated tubulin and gamma tubulin cilia. On the right, you can see the patient-derived fibroblasts have sort of essentially defective-looking cilia, and we can quantitate those as shown below, where we're counting the percentage of cells that have cilia on the left-hand graph, and then we're measuring for the cells that have cilia what the length of these cilia is. And so currently the experiments to rescue this phenotype are ongoing, and we're also working with our collaborators at the University of Iowa to take these into ex vivo models that the university has available, and into some preclinical animal models. So I'll wrap it up there. Hopefully you've got a good feeling for what we're doing at Editas Medicine. We're focused on translating what's a biology revolution into being a therapeutic revolution. With Juno, we've demonstrated effective ex vivo editing of primary human T cells, and we're moving those programs forward to test for functional consequences of that cell editing. And finally, for one of our internal programs that I talked about, the LCA10 CEP290, we've developed functional all-in-one AAV vectors, and we're advancing those into model systems. So I will stop and take any questions, and thank you all for watching, and I'll turn it over to you, Stu.
Okay, thanks, Vic. I was a bit of a incompetent host in that I missed part of my introduction. So I was supposed to tell all the attendees that you can ask questions. And luckily, some of you realized that you could do that even without my having said anything. I was supposed to tell you I would be back after Vic's talk to moderate a Q&A session, so here I am. And you can click on the Q&A button on your screen if you do have any questions. And we do have some already that I'll ask. I was also supposed to say that CD News does not endorse any company products or services that may be mentioned in this presentation or that have been mentioned in this case, and that the talk will be archived and available on demand at the conference website for 3 months after this live broadcast, until December 16th, 2015. So don't go there on December 17th. Okay, so now I'm going to ask a few questions that I have, and feel free to ask more now that I've finally finished my introduction that I was supposed to do before. One question I do have is a couple of gene editing techniques. For example, zinc fingers, TALENs, T-A-L-E-Ns, predate CRISPR, whatever that is, I don't know, exist for, other techniques exist for gene editing. Do they hold any technological or therapeutic advantages over CRISPR-Cas9, Vic?
Okay, I think that's a good question, and of course we get that a lot. I think talons and zinc fingers have been around for longer. People have been working on them longer, so they are a bit closer to being tried in the clinic. So they are a bit ahead of this technology because it's a new technology. I think where we really feel the strength of this technology is in a couple of different places. The first is the easy programmability of the nuclease by virtue of creating multiple guide RNAs. It is incredibly helpful for us early in the process. We can try a large number of different guide RNAs and thereby enzymes to understand what the best combination is to move forward as a therapeutic. So there's a certain flexibility to the system. And a nuance that I didn't go into is there are different isoforms of the enzyme that are mutant in one of the two endonuclease domains. So we have at our disposal what we call nicases. So these are proteins that would cut only one of the two strands. And in the same way we're using two guide RNAs for the CEP290 program, we can program the nuclease to create nicks on opposite sides of the strands, and we can vary both the location and the nature of the kinds of overhangs that we get. And it turns out in data that we've disclosed elsewhere, the ability to create those different overhangs actually has a fundamental effect on how the cell responds to those cuts. And I think those are things that are a bit more difficult to engender with the other technologies, because for you to try multiple proteins, multiple endonucleases, you have to engineer them each individually. And so that becomes sort of a rate-limiting step.
Okay, thanks. Next question is, will the CEP290 targeting be used as a therapeutic or a prophylactic treatment?
The idea is we're going to, sorry, let me start all over again. So for us, the idea is that we'll go into patients who are diagnosed at the molecular level to have the CEP290 mutation, and we'll treat them in the initial clinical trials. We'll treat them sort of in the adolescent stage or in their early teens. We haven't defined this exactly, in order to preserve those photoreceptors. So the initial incarnation is not something that we're going to give to newborns, for example. It is something where we will do a proof of concept in the clinic in older patients to try to preserve their vision for the remainder of their lives. So I guess the answer is therapeutic.
Okay, sorry, we got switched over. All right, how long have you seen the edits stay in the system? Have you monitored long-term knockout of the genes?
So that's a good question. I think in that case, it entirely depends on the turnover of the cell itself. So, for example, in a terminally differentiated cell, such as in the case of a photoreceptor cell, which are not continually turning over, we anticipate that once the cell is edited, it will stay edited for the life of the cell, and presumably the life of the patient. In terms of a rapidly dividing cell, the edits will stay, of course, in the progeny. But if the organ, for example, is rapidly dividing and unedited and edited cells divide at an equal pace, you will see essentially a dilution as an organ grows. For cells that are rapidly turned over, if we don't hit a stem cell or a significant portion of the cells, we'll have to re-administer the drugs.
Okay, that's all the time we have, I'm told. Many thanks to Vic Myer for his presentation, and thanks to all of you for tuning in. There will be more talks beginning at the top of the hour and throughout the day. You can find the full program on the agenda page, located on the upper right-hand side of your screen. In the meantime, please stop by the virtual exhibit hall and the lounge to check out our sponsors and exhibitors, or to network with other attendees. Join the conversation on Twitter with hashtag CENSYM15. Stands for CEN Symposium 15, CENSYM15. Thank you and goodbye.